So, welcome back to Bio 121. We're looking at Chapter 8, and we're thinking about what photosynthesis is. And, again, we're returning to this idea that there's a series of pieces of energy that are traveling, 93 million miles or so to get to Earth and then stimulate subcellular structures that are found inside of plants. And the idea, of course, is we're picking up here as what is light energy, and you need to remember that there's this entire electromagnetic spectrum that's well beyond anything we cover inside this class. It's actually really cool stuff when you think about it, but there's only a small piece of it that we're actually interested in, and that can actually activate the receptors or activate the biochemical pathways required for photosynthesis. But a way to imagine what's happening here, of course, is when you actually look at leaves in the fall and they change their colors. And what's happening, of course, there is the photosensing units are beginning to die off, and those that are remaining are adapting to the wavelengths of energy that are still available when you consider the amount of energy that's available, say, in May versus, say, November. Okay, and when you actually start to see that change, it actually means something. This is what we're looking at here. The visible wavelengths are measured in nanometers, and those nanometers are, of course, if violet has the shortest of all the wavelengths and therefore the most energy, reds have the longest. And what you actually see is this sort of arrangement of receptor units that can actually take advantage of that. And the absorption of light, look, there's a spectrum here. The pigment absorbs a specific wavelength of each of light, and it's unique to the absorption spectrum. And what that means is a pigment itself, chlorophyll A, chlorophyll B, or carotene, a type of carotenoid. Okay, think carotenoid. Okay, and what happens, of course, is these are stimulated. Okay, so chlorophyll A and B capture light for photosynthesis, which is the reason why leaves are green. Okay, green wavelength is what's reflected. Beta carotene helps protect photosystems from dissipating excess energy. So literally it acts as an insulator. These lipid-like structures act as an insulator. Okay, if you think about lycopene for tomatoes and zeaxanthin, which is from the corn seeds, the corn, xanthins, okay, that's the molecule. These literally allow for dissipation of heat to ensure that the tissues aren't damaged. And here's the spectrum, and you can see that there are specific wavelengths that sunlight is most absorbed by. Okay, in this case, the green is what's being reflected, but therefore everything else in the spectrum is what's being absorbed. Okay, so again, here's a sort of thing I want you to go take a look at so you can see how Mother Nature activates all these systems so they can actually function. And the components of the thylakoid membrane are where we want to think about what's going on here, because you actually have photosystems 2 and 1, okay, and these are the sites of light absorption. And then molecules that make an electron transport chain, which means, guess what? We can make ATP, okay? There are 2 enzyme complexes, the NADP reductase as well as ATP synthase. And you notice the NADP reductase is new to our story relative to our mitochondria. So you have photosystem 2 and photosystem 1, okay, and you actually have electron transport chain, which is moving energy from area of charge reduction, right? And therefore, notice what's going to happen. It will pass from light system 2 to light system 1, and you'll see a better cartoon for this in a moment, I believe. Yes, I did put one together. Yay. Okay, and that's what you actually see here. And what's going to happen, of course, now you actually have, here's that thylakoid membrane, here's the thylakoid lumen, here's our ATP synthase, and notice what we're doing. We're transporting hydrogen protons into this space, and yet again, we're creating, using that ATP synthase, the ability for us to attach a phosphate to that ADP plus phosphate here, using the energy of the proton moving down its concentration gradient. But up here, what we're doing is what we're seeing is we are now allowing for the reductase to actually take NADP plus hydrogen protons to make NADPH. And this is significant because now we actually have stored energy. Okay? Now, why would Mother Nature do this? The photosystem 2 and 1 consist of light harvesting complexes. The pigments inside the light harvesting complex pass energy to special chlorophyll A, and then system for photosystem 1 is going to be B. Photosystem A is going to be something chlorophyll A, right? So for photosystem 2. In the reaction center, the light excites the electrons from a chlorophyll. And what's going to do, it's going to pass it to the first electron acceptor of the ETC. And that's when we went back. You can see this right here. Wham! Gets passed there. Okay? Now, what's going to happen? Of course, the lost electron is then replaced. And in photosystem 1, the electron becomes the electron comes from the splitting of water, which releases oxygen as a waste product. So it's photosystem 2, okay, that's going to allow for oxygen to be made. And then in photosystem 1, the electron comes from the electron transport chain. Now, that's a fair bit to remember. What you want to do is you want to come back here and say, oh, I see what's happening here. I have some light energy here, I have some light energy here, and each of these are adding a small piece to a different piece of the story. Okay, and that different piece of the story, notice photosystem 2 that makes oxygen. Photosystem 1 is going to allow for the energy first to make NADPH from NADP plus H+. Now, there are two parts of the electron transport chain in the light reaction. The first, we have this plastoquinone and the cytochrome B6F as well as plastocyanine. Okay, and what's going to happen in this system, the first transports the electron from the photosystem 2 to photosystem 1 via this complex here. So you're literally passing electrons down their electronegativity gradient. Okay, so that means there's something intrinsically different between ETC and photosystem 1. And if you were to think about what Mother Nature might be doing here, is what Mother Nature is doing is she's taking most of the energy, passing it down its energy conduit system, which means photosystem 1 is less electronegative or more electronegative than photosystem 2. Now the final electron acceptor here is what I've already pointed out to you, that NADP plus H plus will yield with that energy the NADPH. Okay? And just like the electron transport system, in respiration, I've already pointed this out to you, down here in the lower part of our cartoon, the ATP synthase uses that gradient to generate ATP through chemiosmosis. We'll probably ask what the major enzyme is of the mechanism of chemiosmosis, and that is ATP synthase. How can it do this? It takes advantage of the hydrogen-proton gradient as a result of the splitting of water. Okay? Now, what's going to happen, of course, is ATP and NADPH from the light-dependent reaction are made in the stroma for use in the Kelvin cycle. So what's going to happen is this energy, notice the NADPH is here, right? And the ATP is here. And notice what's going to happen. You have carbon dioxide coming into the cell. And look, what you're going to make is 3-phosphoglyceraldehyde. Does that sound familiar? That's because by the time we finish producing materials from the Kelvin cycle, we will have produced some form of a sugar from glyceraldehyde 3-phosphate. Okay, and in the process, we break down ATP into ADP, and we use rubiscophosphate, okay, or ribulose bisphosphate, to actually now keep this system going. And it looks a lot like the Krebs cycle, but it's not. It's the Kelvin cycle. And what this means is you're going to fix carbon dioxide, okay, by the enzyme rubisco to generate 2,3-PGA molecules. And in the reduction piece, what's going to happen is ATP and NADPH here, okay, we'll add that electron, say, and now you can actually make glucose. Yay! Okay, and the regeneration continues as you move around. 3 cycles are required to make one GA3P molecule. Okay, so 3 cycles, 1, 2, 3. Okay, for this to actually come to fruition. Okay, yay. Now, here's the Kelvin cycle blown up for you so you can actually see the little pieces. So there's one GA3P molecules, right? So you're going to go around this multiple times, but each step, here's the carbon fixation coming in. That's the exhaling the carbon from your body a plant will pick it up. We're now going to move to stage 2, which is that reduction. Stage 3 is going to be the regeneration, okay, of rubisco. And in doing so, you end up making sure this entire system keeps working forward. Now, there are some Calvin cycle systems which do not result in sugar production. Some plants, like a cactus, like if you're starving to death in the desert, you don't run up to a cactus and eat it. Maybe you'll get some water out of it, but on the whole, it doesn't provide what you would normally need. It provides what it needs to stay alive. And what it's using, of course, is ribulose bisphosphate carboxylase oxygenase. Rubisco is what this is. Rubisco can function as a carboxylase or an oxygenase, where photorespiration does not result in sugar production. Okay? So what they do is they reduce the chances of photorespiration, and in doing so, they ensure that the product that they're going to make is going to be helpful to them. So let's finish this off. Another movie for you guys to watch, you can see that, where you're looking at an animation of the entire process from beginning to end. And what I want you to pay attention to is thinking about the fact that light has to travel a certain number of miles away. Okay, it's traveling at a specific speed. There are unique frequencies that actually cause this entire process to work inside of the leaves of plants. And then what Mother Nature does is she breaks it up depending on the type of plant. Now, this chapter only gave you 2 examples of types of plants. You have to imagine your entire ocean is full of single-celled structures that can take advantage of this. Mountains are of different, depending upon the organism type is what I simply want to say. There are adaptations of this real basic process. And that's where we can finish this. Chapter 8, done.